In our last topic, we talked about microevolution. Micro means small, so we were looking at small changes, specifically changes in allele frequencies within a population over time. Now we're going to take things up a notch. We're going to look at big changes, macro changes. Macroevolution includes speciation. Speciation is the appearance of brand new species, the evolution of brand new species. Spoiler alert, speciation is a product of microevolution. So populations become separate, they start to change, they start to diverge, and if we set up a barrier to gene flow, they become entirely different species, as we'll see. But macroevolution also includes major changes that occur within a taxonomic group. What you're seeing here in this photo is just a random selection of different beetle species. Beetles are fascinating. They are the largest group of organisms. We know of 400,000 different species. So 400,000 species have been characterized. Entomologists, people that study insects, think there might be as many as 2 million. This is a very big, very diverse group. How did we get all of these separate species? That's something we're going to address today. We'll also talk about major innovations, I guess you could say, major changes, the evolution of feathers and the eye and so on. That's something we'll touch on briefly today, and we'll come back to in more detail in later topics. Before we begin today's discussion, we should probably agree on what a species is. And that's a difficult question, more difficult than you might think. Biologists have argued for a long time about what constitutes a species. The definition that is most widely accepted, and perhaps the one that makes the most sense, was presented by Ernst Mayr, a very famous ecologist. He suggested that a species is a population whose members can interbreed to produce viable, fertile offspring. Viable means that they survive, of course, and fertile means that these offspring can have offspring of their own. So essentially what he's suggesting is if we can take two individuals, bring them together, have them mate, and they produce viable fertile offspring, those two individuals belong to the same species. I mentioned this before. Humans and chimpanzees, we recognize them as being different species because we can't interbreed with chimpanzees. Down the bottom here, you're seeing a selection of beetles. They all look quite different, and they all cannot interbreed with each other. So we would consider these separate species. The idea that species are defined by whether or not they can interbreed is known as the biological species concept. Unfortunately, it does have some limitations. What do we do with extinct species? What do we do with microbes that don't have sex? So for extinct species, we only have fossils, and we can't try to mate the fossils, of course. What you're seeing here on the right at the top, we've got two hominid skulls. You can see they're quite different. So one is more robust and it has a crest at the top. And initially, it was thought maybe these are two different species, closely related species. But the current thinking is that this is one species, Australopithecus boisei, and what we're seeing here is a male and a female. But again, dealing with extinct species is tricky. We have to go by morphology. Morphology is form, so we have to just look at the structure, and we have to make some conclusions based only on that. Down the bottom here, you're seeing two bacteria. One you can see is quite spiky, one is not. These are thought to be of the same species. They have slightly different traits, and they're engaged in something quite odd right now. there is a tube connecting them. This is something known as a pilus. It's a hollow tube like a straw, and they can exchange information. This is known as lateral gene transfer. So for bacteria, a lot of them look very similar. They don't have sex, so bacteria reproduce asexually. So kind of hard to apply this concept. And then also they do this really weird thing where they exchange little bits of information. And they can do that with bacteria that look very different and probably are different species. So with bacteria, we rely on DNA sequences, we rely on the shape of the bacteria, their biochemistry, etc., to try to figure out how we should classify them as separate species, even though they don't intermate. Now here's another scenario, a really interesting one, that makes the application of the biological species concept, or any concept of what a species is for that matter, difficult. Here we've got 5 distinct populations, and they're all very closely related. They all came from a common ancestor. We want to try to figure out if they are just populations of the same species, or if maybe they're different species. You can see that the ranges, the geographic ranges of these different organisms are arranged in a ring, and we refer to these as ring species. It's not always an obvious ring like this. This is a best case scenario. Let's start by looking at population number 2. Population number 2 can interbreed freely with population number 3. Likewise, population number 3 can interbreed with number 4. So it's obvious that number 2 and number 3 are the same species, and we might think that number 2 and number 4 are the same species, but they cannot interbreed. 2 and 4 cannot interbreed. What's going on here is number 2 and number 3 are similar enough that they can interbreed and produce viable offspring, the same with 3 and 4, but 2 and 4 have been separated for a long enough time that they have changed sufficiently enough that they can no longer interbreed. Likewise, going the other way, number 2 can interbreed with number 1 can interbreed with number 5, but number 2 cannot interbreed with population number 5. Also, population number 1 can't interbreed with population number 4 or population number 3. So this is a very complicated set of relationships that are going on here that do make the species concept kind of difficult. Remember, in our last topic, we talked about microevolution. That's how populations change over time. If 2 populations become separated from each other and they become genetically isolated, so there's no gene flow between them, then those differences that develop in each population will accumulate, and the 2 populations might become so different that they can no longer interbreed. Once that happens, we have separate species. One species has diverged into 2. We need to have barriers to gene flow in order for this to happen. How does that occur? Well, first of all, we can have the physical separation of 2 different populations. They never come into contact with one another. That's something known as allopatric speciation. Or we could have the populations still occupying the same range and perhaps even coming into contact with each other occasionally, but still diverging because they develop new behaviors, so maybe new mating behaviors, or because they develop other biological barriers to reproduction between the 2 populations. That's known as sympatric speciation. So in this diagram here, we have a pond that's filled with some fish, and you can imagine maybe we have a long period of drought. Maybe we have a couple thousand years where we have reduced rainfall, and this pond dries up and it becomes two ponds. Now we have two populations of fish that cannot physically come into contact with each other. There's no gene flow between them. Those populations are going to change over time due to natural selection, sexual selection, genetic drift, mutation, all of the things we talked about in our last topic. And they can become very, very different from each other. We could have a situation where we don't have the separation of populations physically, but we do have perhaps some fish that maybe start feeding at the bottom of the lake. Some fish come up to the shores of the lake and feed there, and they don't come into contact very often. Those populations could start to diverge. We could have a situation where the fish develop a different coloration and they're not attractive to each other anymore. They develop different mating habits, etc. That would be an example of sympatric speciation. Perhaps the easiest scenario to wrap your brain around is that of allopatric speciation. Let's take a look at a rather extreme hypothetical example. We've got two populations of chipmunk here, and they started out as the same species, but they are now separated by this massive gorge. They would very rarely come into contact with each other. They are physically separated from each other, and that means both populations will accumulate changes. They'll change in different ways. Some of that change is random through genetic drift, but they will change over time and they'll become quite different from each other, perhaps to the point where they can't interbreed at all. So it's kind of sad, really. We've got these poor little chipmunks, and try as they might, they can't intermate. Here we have a similar situation where a geographic barrier, a physical barrier, has separated one population into two populations. On the left, you can see we've got an offshoot of the original population shown in blue that's become separated, become geographically, physically separated from the initial population. It could be that we have mountain building occurring very, very quickly, or perhaps that smaller population just wandered off, migrated off, found its own little isolated valley, and lived there for an extended period of time. In any case, the two populations change, but in this scenario on the left, the populations don't change enough. When they come back into contact with each other, when they find each other again, there hasn't been sufficient change for them to be unable to mate with each other. They mate with each other, the two gene pools recombine, and the two populations basically just reabsorb each other. On the right, we can see a situation where perhaps the populations were separated for a longer period of time, or maybe there was very, very strong selective forces at play. So maybe that new population shown in green found itself in a different environment and there was very strong natural selective forces going on, or there was a lot of genetic drift or whatever. But when those two populations come back together, they are sufficiently different that they can't interbreed. So they're sufficiently different that there is no gene flow between them, and at this point we would classify them as separate, distinct species. Let's take a look now at how barriers to gene flow are created. And I'm not talking about physical barriers now, I'm talking about biological barriers. How, for instance, do sympatric populations, that means populations that have overlapping ranges, how do they maintain reproductive isolation? There's a whole bunch of ways, and we can break this down into two broad categories. We have prezygotic barriers. Those are barriers that prevent the formation of a zygote. Remember, the zygote is the cell that's produced when a sperm and an egg get together. And we can also talk about postzygotic barriers. So these are barriers that will affect organisms after fertilization. Alrighty, let's start by looking at barriers to reproduction that occur prior to fertilization. These would be prezygotic barriers, so they occur prior to the formation of a zygote. They prevent the creation of a zygote. Keep in mind, our goal here is to identify barriers to gene flow. If we have two populations and they're not exchanging alleles, then they can start to diverge from each other. They're not going to share alleles and kind of average out or dilute any of those changes. The first thing we're going to look at is habitat isolation. The classic example is with anoles. Anoles are small, very active little lizards. You find them in the southern US and Central and South America, among other places. I've had these as pets, actually. I had a very large terrarium with quite a few of them. They're really fun to watch. I had one that looked just like this guy here. His name was Elvis. He was kind of a lounge lizard. But anyway, what happens in anoles quite often is they pick different areas in trees. So they have a segregated vertical distribution. We have some anoles that are scurrying around on the ground and catching insects and so on. We have some that will stay on the trunks of trees. We have some that will live much higher up in the trees. There's some that will only occupy the very tops of the trees, the highest point in the canopy. They'll only come down to ground level to mate and lay eggs. So these particular lizards have segregated themselves out. They become adapted to those little tiny habitats, those microhabitats. They become adapted to the slightly different insect species that are found in those areas. And over time, they can change quite a bit. And because they're selecting a very specific zone, they don't come into contact with other populations. And over time, speciation may occur. Let's not leave behind anoles just yet because they are very, very cool. A second barrier to fertilization is behavioral isolation. In many species, we see very intricate mating behaviors, dances, for instance. And if we don't have the proper mating ritual, then we won't get the proper response from a potential mate. Here you're seeing three different species of anole, and they have this structure called a dew flap under their neck. There's a bone in the front that they can stick out, and it extends this flap of skin that's usually quite brightly colored. They also do a little dance. The males do a dance and they shake all around, and they have to do the right dance, and they have to have the right color dew flap with the right pattern on it in order to attract a mate. So once again, you could imagine if we have these populations that are even overlapping, but have developed different mating rituals, then we're going to get an interruption to gene flow. Many species of bird take this even further. So on the left, we have two bird species that are very similar. They have very recently diverged from each other. And in fact, if we were to take the ova, the egg cell from one of these, and the sperm from the other, we could actually combine them and get a viable bird. But these two populations do not interbreed. So we do refer to them as different species. Why don't they interbreed? They don't interbreed because they have a different song. So these populations have developed different songs, and of course the song is used to attract a mate. Mates won't respond to the wrong song. And in many birds, there is a special dance they have to do, as you can see here with these two. There's this elaborate dance that they need to perform, and if it's not performed correctly, mating is not going to occur. If we have populations that develop new dances, they're going to keep to themselves, and that is going to completely cut off gene flow and, of course, allow divergence of these populations to continue. Before continuing, I should point out that in addition to the examples we just talked about with anoles and birds, there are many, many instances of speciation that are currently occurring that have been extensively studied by ecologists. Okay, what's another thing that might prevent fertilization from occurring? What about temporal isolation? Temporal means having to do with time. So here we're talking about two populations that breed at different times in the year. Most animals have specific seasons where they breed. The example on the right here, you can see that we have two populations of a moth, and they breed at different times of year. The top one is breeding in the spring, and the bottom one is breeding in late summer and fall. You can see they also look quite different, and that's because this temporal isolation in their mating season has allowed them to very quickly become genetically isolated. If we have a male from species A, it's not going to be able to find a receptive female of species B during its mating season. So this will very rapidly shut down gene flow and bring about dramatic changes in the two species as they start to diverge and go about in different directions. Now, the next barrier to fertilization is an interesting one, and it's one that applies mostly to insects. This is mechanical isolation, anatomical incompatibility. We're talking about the genitalia here. So the genitals don't fit together in different populations or different species. Now think about what happens in humans. It's pretty straightforward. There's no trouble there. Things line up, they match. But in some insects, their genitalia are so complicated that they simply do not fit together if we're dealing with different populations or different species. Now, without getting too graphic here, I've decided to use the example of an extension cord here. An extension cord has a male end with the bits that poke out and a female end that receives the pokey out bits. And again, genitalia can be more complex than you're used to, and this is something that can actually prevent fertilization. Here's a couple cool examples. On the left, we have a pair of damselflies. Damselflies are very similar to dragonflies. They're a bit smaller and more slender. The one on the top that's blue is the male, and the one underneath is the female. They have a very elaborate courtship ritual with dances and so on that occur in the air. In fact, mating occurs pretty much entirely in the air. These two are just taking a brief break. But you can see that the female has taken the end of her abdomen and shoved it up into the base of the male's abdomen. The male releases sperm at the base of the abdomen, and the female is taking up that sperm. Now, the two bits there have to fit together. There are a number of slots and tabs that need to line up. At the same time, the other thing that's happening, if you look at the end of the male's abdomen, he has a pair of pincers that are pinching the neck of the female. And those pincers have to line up in slots on the female's neck. And that is very specific as well. I think these guys are really quite romantic. You can see that their abdomens, the way they're bent over there, forms a heart shape. Isn't that sweet? Okay, on to our other example here, and this is an interesting example, not really of a process that can cause speciation per se, but coevolution between two different species. We have an orchid there, that's that purple flower, and we have a male bee. Now, the flower is being really sneaky, and this happens with a lot of orchids. That flower is mimicking a female bee of the same species as the male that we see here. So that flower kind of looks like a female bee. The eyesight of the bee is not great. And also that flower is releasing a chemical known as a pheromone. Pheromones will attract mates, and that pheromone smells just like the pheromone that would be released by a female bee. So this male bee comes along and unknowingly tries to mate with the flower. As you can imagine, it's not going to be successful, but the flower is going to deposit some pollen onto the bee. That bee is going to go out and find another female, and chances are he's going to get duped again, and it's going to be another one of these orchids, and that orchid will receive that pollen. So once again, all these structures have to be exactly right. The chemical scent has to be right as well in order for this process to continue. And over time, these orchids become quite dependent, as you might imagine, on these bees. We have one last barrier prior to fertilization. So remember that fertilization is where a sperm cell and an egg cell come together to create a zygote. That's why we also call it a prezygotic barrier. What you're seeing on the left here is a sperm cell that's meeting up with an egg cell. And in step 2, you can see that the sperm is releasing a packet of enzymes. The egg is surrounded by kind of a layer of goo, this sort of gel, and the enzymes will break that down. And then the tip of the sperm cell in step 3 and 4 will find a receptor and bind to it. So there's a protein on the tip of the sperm cell, and it has to have the correct shape in order to lock onto one of these receptors. It's like a lock and key mechanism. The shapes have to match. If they do, then the membrane of the sperm cell will fuse with the membrane of the egg cell, and the nucleus of the sperm cell will get dumped into the egg cell, and then those two nuclei come together, and then we have our diploid zygote. Quite often, we can have very similar species where they might find each other attractive. They might have the same dances and behaviors. They might come together and actually mate, but the sperm and the egg are not compatible. They won't fuse, and this is known as gamete isolation. And this is something that will happen typically with any closely related species if they're separated for a long enough period of time. Those proteins will change, and we get this gamete isolation at play. Now I hear you asking, are there any post-zygotic barriers to reproduction? Is it possible to have a barrier that occurs after fertilization? Well, I'm glad you asked, because yes, absolutely. We can have a situation where we have two individuals come together, they're attracted to each other, they mate, the sperm and the egg come together, the nucleus from the sperm gets dumped into the egg, the nuclei fuse, we get a zygote, but then things go wrong. And this is something known as reduced hybrid viability. This is because the two individuals are not genetically compatible. There's this genetic incompatibility that ends development. So for instance, we might have two populations of mice or two closely related species of mice that have the same information, the same DNA, but it's arranged differently on the chromosomes. This is going to be a problem during cell division. during mitosis. What you're seeing on the right here is the development of a mouse. We might not get that first division of the zygote, or maybe we'll get a few divisions and then problems will arise. So we might get this shutdown that occurs very, very early, so in that top set of photos. Or it's possible we might get all the way to a fetus. But this incompatibility is going to cause issues, and we're not going to get the live birth of a new mouse. We can also have what's called reduced hybrid fertility. This is where two individuals come together and they produce viable offspring. So viable means that the offspring survive, but the offspring that they make are infertile. They can't have offspring of their own. The classic example you're seeing here is with a horse and a donkey. You can take a horse and a donkey and mate them together and you get a mule. And the mule is a healthy animal, but it's infertile. It can't make its own offspring. So what's happening here? If we look at a horse, it's got 64 chromosomes in its somatic cells. And what I mean by somatic, I mean its body cells, its skin cells and muscle cells, etc. So it's got 64 chromosomes. Within the testes and the ovaries, we have a special type of cell division called meiosis, and that makes the gametes, that makes the sperm or the egg. So we have 64. Meiosis cuts the number of chromosomes in half, so the sperm or the egg from the horse will have 32 chromosomes. For the donkey, we start out with 62 chromosomes, and meiosis produces gametes that have 31 chromosomes. In the zygote, those come together, so 32 plus 31 gives us 63 chromosomes. In the mule, we have an odd number of chromosomes. Meiosis doesn't work if you have an odd number of chromosomes. For instance, in humans, we have 46 in our somatic cells. We have 23 chromosomes in our sperm and egg. When the sperm and egg come back together, we get 46, an even number, and then in the next generation meiosis occurs and this repeats. But if you have an odd number of chromosomes like we see in the mule with 63 chromosomes, meiosis cannot occur. We have to have an even number, and meiosis has to produce a whole number, so it doesn't work. The final barrier is something known as hybrid breakdown. This is where you take two individuals, you mate them, you get viable and fertile offspring in the first generation, but in the second generation you see this breakdown. You see a breakdown in viability and a breakdown in fertility. So basically, generation after generation, the organisms get weaker and they get less fertile. An example here is with cotton. So there are a few different species of cotton that are used agriculturally, and as you can imagine, people have tried to interbreed them. They get a first generation, which is okay. Second generation is worse. The third generation is worse still. To sum up, we have several barriers to reproduction, and these are barriers to gene flow. So these barriers will allow populations to diverge and give rise to new species, and they also prevent newly formed species from simply coming back together. So it sets up boundaries between species and keeps species as distinct entities. We have prezygotic barriers. Those occur prior to fertilization, and postzygotic barriers that occur after fertilization. With the prezygotic barriers, just a quick reminder here, we have habitat isolation. This is quite often the starting point in speciation. I used the example with fish before. Imagine fish in a lake. They're competing with each other, so some of them start to specialize in the shore zone and they're living in shallow water. Some of them move off into deep water and feed on what they can find down there. They don't come into contact very often. They don't interbreed. We have behavioral isolation, so we can have populations that stop having a sexual attraction to each other because they develop new rituals, new songs, etc. We have temporal isolation, where we can have populations that develop different mating times. Mechanical isolation, where the genitalia don't line up properly. So again, seen in insects, seen in flowers as well. We have gametic isolation, and this is where even if there is mating, the sperm and the egg won't fuse, and we won't get the combination of the male DNA and the female DNA. For postzygotic, we have reduced hybrid viability, and that's where we typically don't get the birth of the animal at all, or plant. It might fail shortly after birth, but we're not going to get viable offspring. And that's generally due to changes in the way the DNA is distributed on chromosomes. We have reduced hybrid fertility, so we do get individuals developing from a mating that are viable, but they're not fertile. And again, the example we used was with the mule. This is due to an odd number of chromosomes in the offspring. Odd numbers are not good for meiosis. Meiosis does not work with odd numbers of chromosomes, so they can't produce sperm and egg. And then finally, we have hybrid breakdown. This doesn't occur right away, so we can have mating between very closely related species. That's usually what we're seeing here. Species that have recently diverged from each other. They might still attempt to mate. They produce offspring, though, that quickly break down with respect to their viability and also their fertility. Here we've got a very nice example of speciation that is currently ongoing. We're looking at the west coast of the United States. We've got Oregon and California underneath that. And this map is showing us the range of 6 rather distinct looking populations of salamanders. These populations are all very closely related to each other. Whether or not they're all the same species is a more difficult question, but they're definitely all closely related to each other. And it's thought that they can all be traced back to the Oregon population. About 12,000 years ago, the last ice age ended. Ice sheets were retreating, glaciers retreated from this area, and that opened up corridors that the Oregon population could use to migrate south. And as it migrated south, it branched off into these populations. There is a physical barrier that runs down the middle of California. It's a low mountain range, and that divides our populations into a coastal group and also an inland group. And notice that some of the ranges overlap and some of them do not. So for instance, the large blotched salamander at the bottom, it can interbreed potentially with the Monterey population because those populations are sympatric. They overlap. But there is a physical barrier, a geographic barrier between the large blotched and the yellow blotched. Those two species may be deviating from each other through allopatric speciation. The large blotched and the Monterey may also be separating through sympatric speciation. So it's possible that some biological barriers have popped up there. Perhaps because they look different now, they're different colors, maybe they're not attractive to each other. Maybe they've started hunting at different times of day. Maybe they prefer different habitats, etc. So we're definitely seeing a ring species situation here. But as you can imagine, the relationships, for instance, if we pick 2 of them, are they the same species still? Those kind of relationships are quite difficult to establish in a case like this. We've also observed microevolution, for sure, and speciation, most probably, in a laboratory setting. On the right here, you're seeing a classic experiment involving fruit flies from the genus Drosophila. Researchers took a wild type population of fruit fly. Wild type just means the norm for the species. And they took that population and they split it into two distinct populations. One of the populations was given starch as its main food source, and the other population was given maltose, which is a simple sugar, as its main food source. And these two populations were kept separate from each other. They were maintained in different lab rooms. So there was no interbreeding, there was no gene flow between these populations. They grew up, these populations, for decades. This is a very long-term experiment, and it's still ongoing. They found changes in the two populations. The flies that were raised on starch got better at digesting starch, and the flies that were raised on maltose got better at breaking down maltose. So there were some changes in the biochemical pathways in these flies. Genes that coded for appropriate enzymes, they changed. Better copies were favored. That's not too surprising, but what's more surprising is that if they took flies from the starch population and flies from the maltose population and brought them together, there was very, very strong mate preference. So flies from the starch population preferred to mate with other flies from the starch population. And the same for maltose. Maltose individuals preferred mating with other maltose individuals. So over this time, while they were separated, it's possible they developed slightly different behaviors. What you're seeing on the left here are the steps involved in courtship in Drosophila. There is a rather elaborate dance that has to go on. So maybe that changed a bit. It's possible because they've become specialized at different diets, perhaps the pheromones they make that give them a smell that attract the other sex, perhaps those changed a bit. But for whatever reason, we're definitely seeing a strong change in preference. We're not quite at the point where it would completely stop any kind of gene flow, but you can see that we're getting there. An example of allopatric speciation that we've already talked about is shown on the right here. If you remember when we were talking about microevolution, I alluded to how speciation occurs, and I showed you something similar to this. So we have these two populations of mice on an island in Hawaii, and we've got a mountain range that is physically separating them. You can see that their chromosomes have changed during the time that they have been separated. We don't have any gene flow, so we get some random changes occurring. If you look at the mouse on the bottom there and you look at its karyotype, a karyotype is the set of chromosomes it has, you can see one is missing. The missing bits of information, they haven't been lost, they've been transferred to other chromosomes. And there's several translocations that have gone on. A translocation is where a bit of DNA moves from one chromosome to another. Even though they look very similar and they might act the same way and they have the same information, that information has been rearranged, and that's going to make them genetically incompatible. If we brought them together, they might mate, they might even produce a zygote, but that zygote's not going to develop. On the left, we're seeing an example of sympatric speciation. So if populations are sympatric, that means they're overlapping. Here we're seeing caribou in Alaska and Yukon, and you can see there are two distinct ranges. We have this northern range and this southern range of two distinct populations. The animals do look a little bit different, but more importantly, they don't mate with each other. It happens very rarely. So we're not getting much in the way of gene flow, which means these two populations will continue to diverge from each other. Allopatric speciation is probably quite common on chains of islands. We've talked about this example before. This is the example of Darwin's finches on the Galapagos Islands. If you remember, we started out with finches on the mainland of South America, and that's being represented by A here. We have a very small population of those finches finding their way to one of the islands of the Galapagos. This would probably be an unlikely event because the Galapagos are 1,000 kilometers offshore, but we could imagine a rare storm that blows a few birds off course, for instance. Now, once on the island, we've got a new environment, we've got new selective forces. That population is going to diverge from the original population. It's going to change to the point where we would consider it a new species, and we're labeling that as species B. A population of that could then move to another island. Some of the islands are fairly widely separated, and over time, that population, that subpopulation of B, finding itself exposed to new challenges, a new environment, might change into a new species again, species C. Now, if B and C are sufficiently different, and if there's no gene flow between them anymore, then C could move back to the original island and coexist with B as a separate species without any gene flow between them. And you can see this process is repeated over and over again, and this is thought to be the way that we ended up with these different finches living together on the different islands. And even though they were different, of course, we could see that there was a common ancestor due to shared similarities. Darwin's finches, as you can see down the bottom here, differ quite a bit when it comes to the shape and size of the bill. A lot of the differences that have evolved relate to the diet that they eat. But the bodies of the birds and quite a few other structures retain a lot of the characteristics that we saw in the ancestral form. All right, a bit of a summation here about the things we've talked about lately. Remember that microevolution results in changes to the allele frequencies, the genetics of a population. We could have a population that gives rise to a smaller population that then migrates and becomes isolated from the original. And if they become genetically isolated and gene flow stops, then we can get the development of a brand new species. But of course, we have bigger changes that have occurred that we see in the fossil record, that we see by looking at living organisms today. Changes that are much bigger than just a change in the size of a bill. So what we're going to look at briefly now is how we might get those massive differences that separate major taxonomic groups from each other. As mentioned in our very first topic on evolution, kind of the history of evolution, it's important to note that life has to work with what it already has. It never gets a chance to just wipe the slate clean and start from scratch. Evolution builds on what's already there. It's also important to note that very complex structures like the eye don't just suddenly appear all at once. There's several stages, but each of those stages is useful itself. Otherwise, it would not have evolved. So very briefly, a refresher about the evolution of the eye. We started out with organisms that just had a collection of photoreceptors. Those are simply nerve cells that will send off a signal if they absorb a photon of light. Having these patches of pigmented neurons is quite helpful because it tells you if you're in the light or in the dark. That's all it tells you, but that's important information. If you're, let's say, a little invertebrate and you're feeding on algae, you want to be where the algae is. The algae is going to be in the light, but you also might want to seek out the dark for shelter from predators. Now, if we take just that patch of pigmented cells and we bend it into a cup, now we can actually figure out which direction the light is coming from. Can't form an image, but we can figure out which direction the light is coming from. Also pretty useful. If we close the cup over, not completely, but we close it over enough to make a pinhole, we can form a very simple image. And with the development of a cornea and a lens through several transitions, we get an eye that can form a rather good image, something that provides a lot more information for us. Again, all of these steps, all of these intermediates still exist in animals we see today because they all were useful. So it was a process where things evolved by small little steps. Each step proved to be a benefit. If you take a moment to think about it, any change that occurs through macroevolution ultimately has to be the product of a change in development. If we have a structure or an organ that changes over time, that's the result of the development of that organ or structure changing in the embryo and fetus, if we're talking about animals. One of my favorite quotes in all of biology by a man named Lee Van Valen is that evolution is the result of ecology acting on development. And I really like this quote because it ties together a whole bunch of different fields, and it really does a good job of explaining how these fields are interrelated. So evolution, so change over time, is the result of ecology, specifically natural selection. Natural selection is a product of basically weeding out organisms that aren't a good fit for their environment or aren't likely to survive and produce viable offspring. But ultimately that has to act on development. So evolution occurs when we have natural selection, ecology, that selects for new forms of organism. And those new forms of organism are the result of development. What's really important is that development can change quickly. New pathways can develop quickly. And the reason for that is that we have master genes that guide development. So let's think back to giraffes. Giraffes, of course, have a very long neck, but they descended from mammals that didn't, mammals that kind of look similar to a zebra. How did that happen? Well, we had a change in development. For whatever reason, the neck started to develop at a faster rate in the embryo compared to the rest of the giraffe's body. So by the time development was complete, we have this longer neck. Development is controlled by master genes that control a whole bunch of other genes. So there are master genes, for instance, that determine which part of your body will become a neck, which part will become your chest, which part will become your abdomen. For the chest, for example, these master genes are turned on during development, and they turn on all the genes you need to make a heart and the genes you need to make lungs, etc. And they turn off genes that are used in other parts of your body. If we modify master genes just a tiny bit, or we modify the timing of when those genes are turned on and off during development, we can end up with something very different. All mammals, humans, giraffes, rhinos, hippopotamuses, shrews, bats, etc., they all have the same basic body plan. But what happens, what makes these species appear different, is that different parts of the body develop at different rates. Bats have really long fingers that they use to support their wings. The development of the phalanges, the fingers, occurs at an elevated rate during embryonic development. Now, what you're seeing here are some developmental stages in humans on the left and in humans and chimpanzees on the right. Let's take a look at the left here. Now, you probably realize that a newborn baby has an oddly large head compared to the rest of its body. When a baby is born, the head is about 1/4 of their body length. By the time a human is an adult, the head represents about 1/8 of the body length. The reason for this is that the brain and head develop at a rapid rate during embryonic development and development of the fetus. Because the head is developing more rapidly than the rest of the body, the head is quite a bit more prominent. After birth, and especially into adolescence, the developmental rate of the rest of the body is accelerated, and the development of the head slows down to almost nothing. So now the body catches up with the head. So we see this changing of the relative growth rates, the speed of development of different parts of the body. Allometric growth defines the rate of development of a structure related to the rest of the body. And here we have changes in the allometric growth of the head. We see that in the timeline here. Okay, let's take a look at the chimp versus the human. Now, all great apes, that would be humans, orangutans, gorillas, chimpanzees, they have quite a few things in common. One is the number of teeth. We all have 32 teeth as adults and 20 for our primary set. The other things that are similar are the bones of the skull. They're arranged in a similar way, but notice that the chimpanzee fetus, an unborn chimp, and the human fetus look very similar. The shapes of their heads are very similar. Look at the photographs on the right here. At the top, you've got a juvenile chimp, a very, very young chimp. And it has a face a lot like a human face. Compare that to the adult chimp down the bottom. Doesn't look human, doesn't have a human-shaped face. Now, why is that? Well, the chimpanzee head and the human head develop similarly through our embryonic and fetal development, but as the chimpanzee ages after birth, you can see that the jaw grows at an elevated rate. That's what they're trying to show with the grid here. They're showing the relative rates of development. So after birth in the chimpanzee, the growth of the cranium, the part of the skull that contains the brain, slows down, but the growth of the jaw accelerates quite a lot. And if we have a look at what happens in the human situation, the growth of the entire skull slows down, and the entire skull grows at a fairly similar rate. We don't get the big, massive jaws that we see in chimps. Now let's think about this for a second. What this is suggesting is that humans and chimpanzees start out developing at similar rates. The different body parts develop at similar rates. But as we move into adulthood, the chimpanzee growth of the face outpaces what we see in humans. Basically, what I'm trying to say is that adult humans are juvenile apes. You could kind of think of it as we never completely develop into the adult state that we see in other apes. Our development of facial features stops. Changes in the rate or timing of development are referred to as heterochronic changes. So heterochrony is this idea that we can bring about change rather quickly just by turning on a master gene earlier or turning it off later, or just speeding up the rate of development of a particular structure with respect to the rest of the body. We've got an example here with some salamanders. The one on the left is quite good at running along the ground, and it's got fairly long, agile, slender fingers. If we look at a tree-dwelling salamander, it has fingers that are good at grasping onto branches. And the digits, the fingers, are shorter and they're more robust. So what's happened here is just a very small change. A small change in one master gene, or just a change in the timing of that gene, when it's turned on or when it's turned off. So we end up with our ground-dwelling salamander having a longer period of finger development. Whereas for our tree-dwelling salamander, we have a shorter period of finger development. Again, this is relative to the development of the rest of the body. You can imagine that a major change like this could occur rather quickly. Here we have an especially interesting example of heterochrony. This is something known as an axolotl. You might have heard of those before. You can buy them in pet stores sometimes. And it is a type of salamander. Now, salamanders have a juvenile stage, a larval stage, just like we see in a frog or a toad. There's a tadpole stage. The tadpoles have these big feathery gills that stick out from the side of their head, and they use that to breathe underwater. They eventually develop legs. Those feathery gills go away and they develop lungs, and then they crawl up onto land as a land-living salamander. This particular species, the axolotl, does not do that final transition. It starts as a tadpole. It grows legs, as you can see here, but it doesn't lose those gills. It doesn't develop lungs, and it doesn't crawl up onto land. Now, what's really interesting is the sexual development, the development of the reproductive organs and the gonads outpaces the development of the body. When we get to this stage, the body stops developing, the reproductive organs reach maturity, and we end up basically with an adult that's occupying the body of a juvenile. This is something known as paedomorphosis. Paedomorphosis, again, is where you have a sexually mature reproductive individual that actually still has the juvenile form for that group of organisms. Humans are an example of this as well. When we reach sexual maturity, we still resemble the juvenile form of the other great apes, the other members of our group. These Big changes are the result of tiny changes in master genes. Master genes code for proteins known as transcription factors. And what these proteins do is they bind to other genes and either turn them on or turn them off. And one master gene can affect hundreds or even thousands of other genes. What you're seeing in the bottom left here is the evolution of insects. Now, insects have three big body regions. They have a head, they have a thorax, and the thorax is where the legs would attach, and also where the wings would attach if they have wings. And then they have an abdomen behind that. Note that insects are thought to have developed from creatures that have a lot more legs. Think something like a centipede. The mouth parts of something like a grasshopper, they have very complex mouth parts. There's bits all over the place. Those bits are thought to be modified legs. And the antennae are also thought to be modified legs. And appendages on the abdomen have been lost. So the big deal here is that we're starting with a basic body plan and we're just modifying it, changing the appearance of appendages to make antennae, to make mouth parts, or to just keep them as legs. We're losing appendages in some places, but all of this is due to changes in master genes. They have this big effect. We say it's a global effect. That means it affects the whole body. Take a look at the photograph here on the upper right. What you're seeing is the face of a fly. The diagram on the left there, the greenish diagram on the top left, is a normal fly face. And we have these small little appendages which are antennae. But antennae are modified legs. Look at the one on the right there, and that's a very weird fly. Look closely and you'll see it doesn't have antennae on its face, it has legs on its face. That's because the antennae, by default, will develop as legs. All of these appendages, the mouth parts, etc., will by default develop as legs. There is a gene that's turned on just in the antenna, and what it does is it turns off all of the genes that would be needed to make a leg, and it turns on a few extra genes that are needed for the antenna development. So this one master gene can prevent a leg from developing and instead convert that leg into an antenna. And finally, if you look at the last photo there on the bottom right, we have a fly here that has an extra thorax. Again, the thorax is where the wings and the legs should be attached. There's normally just one. In insects, we would have 6 legs attached to the thorax, and then we might have 2 or 4 wings. In flies, we usually have just the 2 wings, but this is a weird fly. It has an extra thorax. It's got a full extra set of wings that we would not expect to see in a fruit fly. Those segments and those body regions are controlled by master genes. So here we have just a small, simple mutation in a master gene, and that has messed up the way these segments are supposed to develop. And we get this major change. We get a segment repeated, and we get something quite different. And before we say goodbye, just a few final concepts here. Speciation is the creation of new species from pre-existing ones, and it's the link between population genetics, so microevolution, and Macroevolution. Remember that speciation is a branching process, and the new species that came from new populations breaking off from the larger population, those new species don't replace the old species. The old species might become extinct, but this is not a linear process. The biological species concept states that a species consists of the largest worldwide population that can interbreed and produce viable, fertile offspring. Species form through allopatric or sympatric speciation. But either way, reproductive barriers have to appear that will keep gene flow from occurring between populations. Those populations remain isolated and there's no gene flow. Even if we bring them back into close contact, then we have separate species. The quickest way to bring about really big changes is to modify the relative rates of development in the body. So to alter allometric growth. And this generally occurs by changes in master genes. Changes in the timing of the genes, when they're turned on and when they're turned off during development, and also changes in where the master genes are activated. What organs are they activated in? Evolution is unpredictable. It's the product of changes in the environment, changes in other species, chance mutations, and luck. But remember, evolution is not a random process. A lot of people incorrectly state that it is. They'll say things like, how can something complex evolve through random chance? It's not random chance, because natural selection is going to select for traits that are beneficial. So we can get a progression that occurs. We can get a trend that occurs. The development of the eye, the development of wings, and so on, where we're building up little tiny changes one on top of the other because each little change is helpful. And that is not a random process. Now I'd like to sum up everything we've learned about evolution so far. So to sum up the first three topics that we've looked at, natural selection favors genetically based traits and behaviors that enhance reproductive success. Individuals that leave behind more offspring will contribute more alleles to the next generation. But keep in mind that allele frequencies can also change in a non-directed or random way due to genetic drift. Speciation occurs when populations become genetically isolated long enough for reproductive barriers to develop. That maintains the separation of species. Changes in master genes and the timing of development can bring about really rapid macroevolution, and that's the little jumps in evolutionary rate that we see evidence of in the fossil record. And finally, here's some helpful terminology. Now, we've spent a lot of time talking about evolution. We'll come back to these concepts over and over again in the course when we look at ecology and when we look at the diversity of life on our planet. So until next time, cheers.